Sweet. Welcome everybody to Sustainable Design Masterclass. I'm Neil Spackman. You can also see it says Wayne Doorband next to his head, but this is Raleigh Latham, and together we are the co-founders of what we lovingly call the Sustainable Design Masterclass. We started about 2 years running webinars focused on the interplay between entrepreneurship, sustainability, and agriculture. Some of you might be familiar with my work in Saudi Arabia, but we try to have luminaries on in those regions like Elaine. We're coming up on our 40th webinar in the 2 years we've been doing this. And all of you had to sign up on our list to get here. So you will be getting our emails when we send up the recording for this. We tend to keep all of our webinars free for at least 72 hours after we do them. So if you have to duck out, if it's very late at night where you are, you're still going to get the replay for this. So welcome. We're really happy to have you all on. Hope you can find some of our other webinars just as interesting as this one. And I'm going to turn it over to Raleigh.
Hey there, everybody. Stoked to have you on. I'm looking at all the names. We got about 300 people on live right now, but I'm sure by the end of it, who knows, there might be 700 people, which is just amazing. Does that mean you guys are as into soil restoration and ecosystem restoration as I am, which is phenomenal to see. So I want to tell you about the webinar we've got put on today. So our webinar is called Extreme Climate Restoration Using Soil Microbiology to Restore Farms, Landscapes, and Ranches. This is going to be amazing. Elaine's going to go on for about an hour and a half on the topic. So you want to be present for this. So I'm going to ask you, turn off your cell phones, turn off Facebook, you know, shout at the mailman, say, hey, get away from my house. I got a webinar to listen to. You know, lock your kids in a small room, give them some nutritious grass-fed beef and a glass of water, because you want to take this all in, because this is really amazing, important information. There's going to be a lot of it flying at you, so you don't want to be all distracted. So first of all, we're going to have our presentation with Elaine, and then right after, our guest Vivian's going to tell us about her experience as a student learning from Elaine. She's going to tell you about an incredible opportunity to learn from Elaine directly, and to top it off, one of you, I got 2,000 entries for our little contest giveaway. One of you is going to win out of this hat. I narrowed it down to 2 names from 2,000. As people from around the world, from Hopi elders to people with 1,500 acres. It was just amazing. So I'll be really excited to reveal the winner of that. And oh yeah, and after the webinar, I'll send you the replay to everyone, and everyone's going to get a free 1-week access to Sustainable Design Masterclass. Hooray! So without further ado, I'm going to switch this over to Vivian. Hey, Vivian.
Hello, and thank you for the enthusiasm, Raleigh, and your introduction. Hello, everyone. Nice to meet you all. My name is Vivian Kalouxilos, and I am in Quebec, in Canada. And I'm here to introduce a woman who's helped me so much in my life and has taught me so much, Dr. Elaine Ingham. She is the founder of Soil Food Web, Inc., and the co-founder and head of research of Environment Celebration Institute, and one of the world's top soil ecologists, microbiologists, and a woman who is really offering something accessible, concrete, and revolutionary to agriculture and all land management practices. And also, most interestingly, the connection people are making is with the climate. And so we're honored to be here and with Elaine, to have her in person. Thank you so much for spending your time here, and we're very grateful to have you with us. It's a very special day, so I hope you all enjoy.
All right, thank you, Vivian. So yeah, Elaine, you just got to enable your screen, and then we will be good to go. Awesome.
Is that it?
Oh, yeah, why don't we go back to the start? There you go. So just click the View Slideshow, and then we'll be all set.
Okay, hold on. Yeah, I'm not sure exactly where to go and what to do. So,
The webinar's ruined everyone. No, we're fine. Usually you go to slideshow, and then it's like you click that in that bar, and then you start, click from the beginning. Oh, it showed up for 1 second there.
yeah, because it should be show screen.
Yeah. Well, what we're seeing right now is your PowerPoint. We're seeing PowerPoint open.
What I'd like to have up there, and
Okay.
man, it is not happening on my screen.
Okay.
So,
Great. So now you're gonna go.
Is it there?
Bam! I can see it. I can see it. Hey, everybody, let us know. Give us some ones if you can see Elaine's presentation.
Hopefully, it's coming across for everybody.
Okay, got a lot of ones. Maryland, MK, 111, looking good. It's up, good. And everyone can hear Elaine? Can everyone hear her coming in all right? Yes.
Am I loud enough?
Go. Awesome.
Okay, we're ready to roll.
All right, we are ready to roll. So, I'll hand it over to you, Elaine. Thanks again.
Okay, thank you, Ellie. I'm going to concentrate today on very practical things. Of course, I have to start with the background. We are trying to put the soil food web, all of these different organisms that comprise the soil food web, back into your soil. Because the decisions we make in agriculture are to disturb and till too much. It'd be fine if we till just once a year, but we don't do that. We often till up to 14 times in agricultural systems, maybe even more than that at times. And every time we till, we're crushing, we're destroying, we're slicing and dicing all of these organisms in the soil. And as we lose these organisms, we lose all the benefits. All the things that nature has developed over the last billion plus years to grow plants in the soil. And of course, your plants came into existence, rooted plants, came into existence after all of these organisms were present and functioning. And so, of course, the plant didn't, also put into its DNA all the enzymatic abilities and all the nutrient cycling that all of these organisms were doing in your soil. They didn't have to, because these organisms were already there. They were already doing all this work. So the plant is very intimately connected with all of these organisms in the soil. And really, through the course of my career, we've only begun to understand all of these interactions. So we have a long way to go. We don't know everything yet by a long shot, but we certainly can stop using all those toxic chemicals that the Green Revolution told us is the only way that we can grow enough food to keep the planet, to keep human beings fed on this planet. And that's absolutely not the case. Usually what we see is, as the conventional is not a great term, it's not conventional by any means, but that's what we call it. When conventional agriculture starts to destroy all of these organisms, and basically you're left with nothing but bacteria, and bacteria alone, they cannot perform all the other functions that all the rest of this food web has to perform in order to benefit your plant in all the ways that's possible for these organisms to benefit the plant. So once we're down to just bacteria in our agricultural soils, then we have to use inorganic fertilizers. We have to use pesticides. But that's so opposite the way nature designed the system to work, that slowly but surely we're destroying the ability of our plants to even have the nutrients in them that they're supposed to have. So how do we reverse this? I don't want to spend a lot of time on, the gloom and doom stories. I'm sure you're all just sick to death of hearing about all the ways that things are going bad. How do we reverse that? How do we get this biology back into the soil so we aren't dependent on any of those toxic chemicals? We don't need inorganic fertilizers. So let me quickly summarize that. First of all, of course, we're going to start with sunlight. Sunlight is energy coming to the planet's surface, and photosynthetic organisms, of which plants are just the most recent to arrive on this planet. But as that sunlight and the plant then takes up CO2, the plant is going to release this oxygen, the O2, and it's going to bind one carbon from the first carbon dioxide to a carbon from the second carbon dioxide. And now it's starting, that plant, through the process of photosynthesis, starts to make a chain of carbons. And that's what we call sugar. Those sugars, you know, how long is that sugar going to be? How many branches is that sugar going to have? How complex? Well, it depends on what the plant is and where the plant grows and all these other factors of environment. But all plants will then take that sugar and send it down into the root system, because that's where the sugar has to pick up all the other nutrients that that plant requires. Plants can't grow on sugar alone, just like we can't grow on sugar alone. So how do they get the nitrogen, the phosphorus, the sulfur, the magnesium, the calcium, all of those nutrients? Well, they're in the rocks. Those nutrients are in the pebbles. They're in your bedrock. They're in the boulders. And as your parent material, the boulders, the rocks, break down into the different size fractions of sand, silt, and clay, those nutrients are very much available to the right enzymes made by, oh surprise, bacteria and fungi. Bacteria make one certain set of enzymes to pull nutrients off the surfaces of the sand, the silt, the clay, and of course off of from the organic matter that might be in the soil as well. Fungi make a whole different set of enzymes that will pull different nutrients, and in both instances those nutrients end up in the fungal biomass or in the bacterial biomass. One of the things these bacteria and fungi have to have is food to do this work. And so the plant is putting out exudates to grow the specific bacteria that will make the specific enzymes to pull the nutrients from the sand, the silt, the clay, the rocks, the pebbles, the organic matter. And so there is no soil on this planet that lacks the nutrients to grow plants. It's just that in the conventional world, they ignore that total set of nutrients that are present in plant not available forms. Instead, they concentrate just on the soluble nutrients. And if you kill the bacteria and the fungi off, you can't get soluble nutrients back into your soil. So what are you going to use to grow your plants if this nutrient cycling system established by nature, and that has been functioning for the last 4 billion years on this planet, what if you kill all these organisms off? Then how are your plants going to get the soluble nutrients that they require? And that's where we think we have to put on inorganic fertilizers. Well, even that falls apart when you think about, it's not just nitrogen, it's not just phosphorus that your plant requires, it's not just calcium. Everything has to come from the soil. It's the boron, it's the zinc, it's the selenium, it's all of those micronutrients. In whatever concentration they're needed by the plant's got to take that up into its body through that root system. And so we've got to have this nutrient cycling system. Well, if you were listening, you realize that I only got to the part where the bacteria making enzymes, as directed by the plant, by the exudates coming out of that root system. The bacteria are going to make those exudates. The root is going to make the exudate. Bacteria are going to eat those exudates, and that promotes the growth of those bacteria that make the certain enzymes to pull the certain set of nutrients from the sand, silt, and clay present in that soil. Whereas a different set of exudates are going to be made for fungi when the plant needs those other nutrients that fungi go after, and their enzymes pull those nutrients into the bacteria or the fungi, and those nutrients are now in the bacteria and fungi. Well, that's not available to your plant. So how do we get those nutrients out of the fungal biomass, out of the bacterial biomass, and into a form that your plants can take up? That's where these next set of organisms, the third trophic level, have to be present in that soil, because protozoa eat bacteria. And so as the protozoa do their job of eating the bacteria, the concentration of nutrients in the protozoa is much lower, much less than the concentration of nutrients in the bacteria. So when a protozoan eats a bacterium, the excess nutrients are released in a plant available form. When we have bacterial feeding nematodes eating bacteria, exactly the same thing happens. The nutrient concentration required by nematodes is so much less than what a bacterium has in its biomass that when a nematode eats a bacterium, all those excess nutrients are going to be released in a plant available form. And of course, your plant can now take those nutrients up if it needs them. If your plant doesn't need them, the plant doesn't take it up, and then quite happily, the bacteria and fungi that are in that root system take up whatever the plant doesn't need, holds on to all those nutrients, it keeps them from leaching. So see, we don't have the loss of nutrients from our soils when we have this biological system in place. We don't have the damage to rivers and lakes and streams and groundwater that we're seeing when you put on a soluble inorganic nutrient onto the surface of the soil. So we want to get this nutrient cycling system reestablished. I didn't talk about fungi. What eats fungi? So fungal feeding nematodes eat fungi, and the concentration of nutrients in fungi is so much higher than in what eats it. So microarthropods the same way. When the fungus is eaten by either of its predators, soluble nutrients are released, and that's now available to your plant. The plant takes up what it needs. So see this connection? So extremely important, but that's why we don't need inorganic fertilizers. It's absolutely crazy that we're out there paying money to put on additional nutrients that are already present in your soil. If you've killed the biology, then your plant cannot access those nutrients which are present in your soil. If you get that biology back into the soil, then stop putting on inorganic fertilizers. It's not required. And so time after time, as we reestablish this biology in the soil, people stop putting on the inorganic fertilizers. And every once in a while, they'll kind of test the system. Do they really need, not going to get any benefit out of those inorganic fertilizers? So they put some inorganic fertilizers back on. And what they find is that there's no benefit to the plant. The plant does not start to grow better. So there's kind of a test. Do you have good biology in your soil or not? Put on some inorganic fertilizers, and if your plants grow better, that's Mother Nature telling you you don't have the food web in your soil that you need to have. And so when we put that inorganic fertilizer on, there's another problem that you need to understand as well. If you're putting on less than 100 pounds of inorganic fertilizer on your soil during one application, that's not really going to affect the biology too much below ground. But if you're putting anything more than 100 pounds per acre, you're going to be killing these organisms that are in the soil, because every inorganic fertilizer is a salt. And that salt takes water away from the fungi, away from the bacteria, from the protozoa, nematodes, microarthropods, the higher level predators, and it kills them. And so again, how can you make those nutrients which are in your soil available back to your plant if you just killed most of the organisms, the bacteria, the fungi, protozoa, nematodes, microarthropods, if you just killed them? Then you think that you have to put on more inorganic fertilizer. And because that kills more of this biology in the soil, whatever little is left, then the next time you think you have to put on even more inorganic fertilizer. And when you think about that situation where we have, over the time since we started putting out the Green Revolution recommendations, from that time we've had to put more on inorganic fertilizer. The next year we have to put more on, and the next year it's even more. And isn't that the definition of a moron farmer? We've got to get away from that. We have to understand what we're doing. It's not the fault of the farmer, really, that they keep putting more on and more on and more on. It's the fault of our educational system. They haven't been looking at this. They haven't understood how this biology in the soil works. And that if we get this biology back, we don't need those inorganic fertilizers. We don't need those pesticides either. Because when we start thinking about disease around those root systems or around the top part of the plant, the protective layer around the surfaces of every part of that plant are these microorganisms. When that plant puts out these exudates that it's making from sunlight and carbon dioxide, so there is no shortage of that in the body of your plant. What's lacking are all those other nutrients. But as those exudates come out into that soil around that plant, that means all these bacteria and the fungi, the beneficials, the good guys, are now protecting your root system against any pathogen, against any pest. When we put on an inorganic fertilizer, when we put on a pesticide, we're killing those organisms. And now the bad guys can attack those root systems and nobody's going to object. There is no protection left on that plant. And so open infection courts, diseases very easily get in. There's no one to protect. And so now we think that we have to have the pesticides because look at how easily these pathogens and pests attack our plants that are growing so well. Well, the fact is they're not growing so well. They're taking up soluble inorganic nutrients, yes, but those nutrients are unbalanced. There's way too much nitrogen getting into the plant. There's way too much phosphorus, depending on what chemical fertilizer was applied, and your plant is having to consume those nutrients, and therefore the tissue in that plant is unbalanced, and therefore very easily attacked by these pests and pathogens and problem organisms. So if we are getting this biology back into the soil, back in coverage, how do the organisms in the soil get onto your leaf surfaces? Well, that's the job of some of these bigger organisms that move both from the soil onto the plant surface. So we've got to get all of these benefits. So a healthy food web does all of these things. It suppresses diseases because of that protective layer all over the surface of the plant. Out into the soil, these organisms are building soil structure. So building the microaggregates, the macroaggregates, so you hold more water in your soil. So we don't have drought problems. We increase that water holding capacity. Now the roots can grow deeper down into the soil and reach that groundwater. It can reach that subsurface water. And so we've got to build that structure so we suppress the diseases, which for the most part, the disease-causing organisms require reduced oxygen conditions. If you have fully aerobic conditions, the aerobic organisms, the beneficials, grow. And it's not until you reduce oxygen, compact the soil, waterlog the soil, oxygen cannot move in there, and now the diseases win. That's the perfect environment for them to grow in. So with the proper biology, I explained how we retain nutrients, so we don't have these toxic chemicals or the salts moving into our water systems. We're going to make nutrients available at the rates the plant requires because the plant's in control. Let the plant be in control of its own life. When we are making certain that all of the nutrients going into your plant are properly balanced, everything is available to your plant, then the flavor and the nutrition. Well, nutrition is flavor. When you take a bite out of a really well-balanced, fully nutrition concentration of nutrients inside that tomato, it tastes incredible. What makes me really sad is when you hand a small child a beautiful, wonderful, luscious tomato, and they take a bite and they go, whoa, that's kind of strong, isn't it? They've never tasted a good tomato. The tomatoes they've had have always been lacking in some nutrients, so they've never experienced the proper flavor. What does that say about the health of that child? So this works its way. The soil and what's in it moves all the way through our society. We've got to understand this. And so, like to point out one of the first papers in the scientific literature that really talked about the role and the function, where there were actual measurements made with how much more nitrogen ended up in a plant when this food web was in place and functioning as compared to dirt, where there were just bacteria and nothing else present in that dirt. And really, we have to understand that soil requires, you cannot be dealing with soil if you don't have all the microorganisms. That food web has to be in place for nutrient cycling to happen. It's not soil if there's not life in that soil. It's not soil if you don't have organic matter. So if you've ever bought engineered soil, you should just laugh hysterically, because all that's in an engineered soil is sand, silt, and clay. There's no organic matter. There is no life, because all of that sand, silt, and clay was sterilized before they put it together into that supposed soil. So no wonder it's hard to grow plants in that kind of material. So this is the first paper. It's Ingham et al. Back in 1986, an ecological monograph. So really long. Most scientific articles, you'll notice, are about 3 pages long. This is about 25, 30 pages long. And it goes through what happens when you have just bacteria or just fungi. Plants die. If you have bacteria and fungi, plants die. If you have bacteria and protozoa, the plants do okay. They're getting some nutrients cycled. If you have fungi and fungal feeding nematodes, then the plant's going to live. But if you put in the bacteria and the fungi and the protozoa and the nematodes and the microarthropods, now the plants flourish. Incredibly more productive. So we want to talk about some specific examples of restoring soil biology on landscapes. So here's an example of East Coast city, Boston to be specific. And through the growing season, you can see the differences in the lawn. So look at the circled lawn in the upper right-hand area, and you can see how green that lawn is, even though this is middle of September and a year of drought. None of these folks could be watering their lawns on a regular basis. They could only water, and it was like on Wednesdays and Sundays or something like that, and they could only put out half an inch of water. And so look at all their lawns. These are the people that are on ChemLawn. They had a chemical fertilizer company that would come out and put on, every so often, put on the chemicals and say that, that's the best they could do, and look at their lawns. What are the green plants growing in these lawns? It's the grass that's brown and, not growing. It's dormant. Yes, it will come back, so it's not dead. But look what plants are winning, the weeds. And so you look at this lawn, everything's bright green. Oh, well, they've been watering constantly. Why haven't the water police come and arrested them? Well, a year ago, their lawn, late in September, looked exactly like everybody else's lawn because they used ChemLawn. And so what did they do between last year when they were on the ChemLawn system, between then and now? What did they do to have this beautiful green lawn? And look at their trees, no diseases, their shrubs, their vegetables in the back garden. No diseases, no pests, no problem organisms, no need to put on inorganic fertilizers, and even more, you don't have to be watering. These folks have not watered this lawn, they have not irrigated this lawn since a year ago, October. That's when we started to assess the biology in their soil, and so that's always the first step. What do you have there to begin with? And then we're going to put into the lawn, into the area around the shrubs and the trees and the vegetables in the backyard, we're going to make certain we get the proper balance of the bacteria and fungi, protozoa and nematodes. Make sure that we're going to get those organisms back in there. And so a year ago in the fall, applied approximately 50 pounds of compost to the yard. And then around the trees, we added compost that was a little bit more fungal, because trees require more fungal biomass. They require ammonium more than they require nitrate. And it is fungi and ammonium that are very much linked. And if you take the life in the soil classes, we'll go through all that chemistry with you and explain. I just don't really have time now. And then around the shrubs, not quite as fungal, so not as many fungal foods, lower inoculum. In the lawn, made sure that it was an equal biomass of fungi to bacteria, because that's what productive grasses require. And then for the vegetables, the tomatoes, the potatoes, the celery, the lettuce, cucumbers, all of that, the proper ratios of the bacteria and fungi all winter long. These organisms grew in the soil, lots of organic matter, and they balanced things out, built structure, put themselves, grew all over those root systems, so those root systems were well protected. So from that soil, all the other creatures, the birds, the mice, the lizards, the whatever, earthworms, everything, those organisms that will crawl up the trunk and inoculate all of the leaves as they bud out. So all of the above ground part of the plant was protected from those diseases and pests. Look at the trees in the neighbors' lawns. Look at the bare branches. These trees are all sick. They're unhealthy. They don't have the nutrients. They're all being attacked by caterpillars or aphids or whiteflies. All of the street trees, all of the shrubs, all of the plants, they're showing signs of diseases, lack of fertility. They're being attacked by pests. And of course, what's ChemLaw's answer to that? Well, they're all going to come out with a toxic chemical and kill those organisms, which is not the right way to handle this. You're not reestablishing, you're not fixing anything. You're just walking down that pathway to hell when we're doing these things. We don't understand what those toxic chemicals, all of the damage that those toxic chemicals are causing in these systems. Whereas if we just start getting the right biology out here, get it established. So no irrigation of this lawn at all. Peter would say that he would stand in his window at 2 a.m., 3 a.m. in the morning, and he would look across here and he could see the police sitting there in their car. Who knows how long? But because the ticket they could write, if they could catch him watering his lawn, they could write a hefty ticket, but they could never do that because he wasn't watering his lawn. Because the structure in the soil, the organic matter that had been built, the biology going deep, the root systems on his grasses were going down 6 to 8 feet in that first growing system. As time goes on, those root systems grow deeper and deeper. So these are the kinds of conversions that we want to help people achieve. And that means you've got to be able to monitor the biology in the soil. You have to learn how to use a microscope. You have to do shadowing, microscopy. We can teach you how to do that in one day. Or you start working with one of our advisors, and they do that work for you. So whether it's on a landscape like this or a landscape like this, so this is work that we did with a company called Sustainable Growth Texas, and I've trained many of their people. They know how to do the microbiology. This is the pipeline that went in, a gas pipeline. So they dug up all of this area between these two fences. They dug all of that up, buried the pipeline, covered it back up. But this is sugar sand. This blows with the wind. This is after the first rainfall on this area, after they covered. And look at what's happening already. We already have erosion. starting to take away the cover on the gas pipeline. How soon is it? How many rainfalls do we have to have before this gas pipeline becomes uncovered? Because they reseeded it. They put in seeding, and there was a time not too long ago, before this picture, where this was all nice and green all the way along. It looked like this area here. But look what's happened with the wind and with the rain. The only place that's being retained is just in the fence line. Yeah, there's a little bit of grass growing, but it's not going to make it, is it? All the hydroseeding is landed down there in the creek bed. And so where is all of that seed and all of this dyed woody material going to end up? Not here, not doing its job. So Sustainable Growth Texas, Betsy Ross is the person who runs it. She came out and said, okay, the way we're going to fix this is put out a nice inch and a half layer, maybe 2 inches of compost with all the right biology in here. We're going to come out with a compostee, and we're going to apply another application of those organisms if it's needed in order to establish this biology in the soil. And they mixed the seeds of the grasses that they wanted, that the people owned, the surrounding areas wanted to see. And so all of that went in, and here we are 6 weeks later. Completely covered with grasses. Yeah, there's a few weeds. Yeah, so okay. Come out with your biology. Apply just to these spot areas where you've got weeds growing, and the weeds will be selected against, and the good guys, the grasses that you want, will be promoted. They will be selected for, because the beneficial, the really highly productive plants require this equal biomass of fungi to bacteria, whereas the weeds require 2 times more bacteria than fungi, or a fungal to bacterial ratio of about 0.5. You know, as soon as they realized that there was this much grass in this area, what did they start to do? They started putting in the water pipes to bring water to the water troughs for the animals. And they put cattle out here and started grazing cattle. Whereas, would we ever have been able to graze cattle on this? And all it took was 6 weeks. If you understand the biology, if you get those organisms into the system, if you make compost in the proper way, that will get that biology back out in the system. So going to larger, people always say, well, those are such small areas. Can you really do this in larger? Yes, we can. So we look at, this is an example in dairy farms in Australia. And the farm that we started working with was the Whitings on the left-hand side, and then the neighbors that stayed in conventional pesticides and inorganic fertilizers going on the meadow, on the grasslands, pastures on the left, on the right-hand side. So the Whitings on the left, the conventional on the right. A year previous, we had put out compost into these pastures before fall and got that really good biology growing, watched that fungal component come right up in these soils. So come the next springtime, they did not, the Whitings did not have to resow this pasture. They thought they were going to have to, but because of the improvement in the biology, they didn't have to pay that $20,000 to do the resowing on any of their pastures. This is a 300-acre dairy farm, and there's always a certain number that they had to resow every spring. Well, they saved that money. So right from the very beginning, they spent maybe $500 total, putting the biology out, getting the biology out, making their own compost, which they made from their own waste materials from their own farm, which saved them $15,000 because they were no longer being fined by the EPA because they had manure lagoons. So huge savings all around, and that's just in the first 6 months. On the other side of the fence line here, the conventional farm, they had to resow their pasture because they just didn't have enough grass growing in there. And you can see the tillage lines where they tilled in that grass seed to get it in contact. And notice that that grass is not growing to speak of. So they grazed only once. And you have to realize this is Australia, so the beginning of the growing season is in November. So back in October is where we put on the compost on the whiting side, whereas they resowed the paddock over on the conventional side. And so the whitings have saved all this money, the neighbors have not. The whitings have been able to graze 5 times. Now this is late in the growing season, this is fall, and so the whitings have grazed their entire herd on this pasture at least 5 times, and the last grazing that they did was 10 days ago. And look at how much the grass has grown. On the neighbor's side, even though they resowed this pasture in the spring, back in October, they have gotten no growth. They've not been able to put their whole herd. They've only been able to put sick animals on that pasture because there's not enough grass growing. In the first year that we work with growers, with dairy people, we save them on a 300-acre dairy. We save them on average $200,000. So you can see where this has had a big impact in Australia with farmers, with the dairymen who choose to go this direction. And there's some really clever ways to band together and make compost that ends up with you not having to buy a turner or the water wagon, that you don't have to be making your own compost. It's done for you on your own land. So this is the pasture in May. So this is a month later. This is definitely into the beginning of winter in this part of the world. Yeah, those are eucalyptus trees. They don't lose their leaves in the background. No nitrogen has been applied on this pasture since the compost addition back last October. And the pasture's ready to be grazed 10 days after the last grazing. These farmers increased their herd size. They didn't have problems with calving. Their milk is rated the highest because it's got the most milk fat, so they get the greatest money for their milk. They don't have to be put on inorganic fertilizers. They don't have to put on the pesticides. They don't have the white flies. They don't have all of the pests and problems, and the parasites have been reduced. They say to me that if they had just known that the reduction in their veterinarian bill would drop so much, it more than paid for all of the work that we did getting the compost and the compost extracts out here on this property. They've made so much money. So you have to understand what your crop requires, what was in your soil to begin with, and we've got to take that, well, it's really dirt when we start with most people, and what do we have to do to get the biology back into that soil to call it soil. And it's very doable in one growing season if you have the compost. So we've got to get that source of compost. We can work with you, help you. Usually we can get people to the point where they've got the biology in that compost, but it takes 6 months, it takes 8 months to really do that job. So if you can have a neighbor that's already started this program and is already making this great compost, you have a blessing, because you don't have to make that compost. You just ask them for some of theirs, or they help you. They bring the inoculum. They improve, reduce the time that it takes to get this biology that you need back into your soil. This is an example of an area working with Hendrika Straven, where part of the pasture, grassland, was treated conventionally. The other half, we went in and we made certain to till this really good compost in. It's what Hendrika Straven calls his essential soil. So he puts in this mix, tilled it in the last fall, because the compaction in that pasture was just so horrific. Conventional agriculture, conventional applications make the compaction worse. We have to come in, often if we want this instantaneous kind of turnaround, we've got to come in with a tillage event. And so in this particular instance, we key lined in. So it's not disturbing. It's not like coming in with a chisel plow or a disc plow or moldboard or rototiller, which completely and totally disturb the soil and slice and dice and crush all of our beneficial organisms. So with the key line plow, it's a small disturbance. Your tines on your key line plow, or yeoman's plow is another name for it, are about 6 feet apart. We have compost tea or compost extract going into the soil on the back side of the stanchions. And so to all the breakages and all of the breaking of those compaction layers, we are putting out the proper biology down to a 4-foot depth. So here's the seed was then planted on July 15th after all the preparation of the fields was done. And the ryegrass was treated by soaking it in a compost tea, so we would have the organisms on the surface of the seed. So as soon as the seed germinated and started to grow, the biology would be on the roots, it would be on the stems. You're protecting your plant. Diseases are not to be worried about. So through that growing season, we applied the compost tea compost tea once, sorry. And then on November 6th, we took a 10-inch diameter PVC pipe, pounded it into the ground. I should say Hendrikus did this work, not me. I stood and watched. I supervised. Pounded the pipe into the ground, then pulled it up. Yes, you have to have some pretty big equipment to pull that 10-inch diameter, 4-foot depth PVC pipe that you pounded into the ground. Pull it up out of the ground. We then sliced the PVC pipe off. We used a hose to wash away some of that excess soil. And here's what you see. The root systems on this grass are down 4.5 feet. And that's in 3.5 months of growing. How deep do root systems of grasses go? They ought to be going down at least 4.5 feet in that very first growing season, especially if you want to stop applying water. If you don't want to water your lawn anymore, this is what you need. You've got to build the structure. The organisms have to be present to do the nutrient cycling. So very happy plants. I'd like to point out that we cut this this plant, the plants that you can see we cut the whole pasture, but we cut the plants to 1/2 inch tall. You can see they're already growing back. 3 days later, they're already growing back. You can't be sloughing your root system if you want grasses to grow back. We need to. They have to keep this root system. But then it's not a stress on this plant when you mow it or when your animals eat it. Because look at that root system that is immediately going to start pulling more nutrients into that root system in order to grow the above ground part of the plant instantaneously, and then start pumping those sugars back down into that root system. No weeds, no diseases, no need to put on inorganic fertilizers, no needs for pesticides. It's not going to be very expensive for you to grow plants. And maybe that's also where we're trying to get. As we grow these plants, we're going to be increasing the organic matter. If the root systems of these plants aren't going just 2 or 3 inches into the soil, but they're going down 4, 8, 12, 20, 25 feet into the soil, depending on which plant you have, and wouldn't you want to pick the most productive? If you pick the most productive plants, then they're going to be putting their root systems way down, and aren't we going to be sequestering more carbon in our soil? If you want to take all that elevated CO2 that is in the atmosphere, you want to take out the methane, you want to take out those greenhouse gases, we have to grow more plants. Well, but do we actually have to cover more of the Earth's surface with plants? Or if we get the root systems growing down and we don't have to use all these toxic chemicals, if we weren't making inorganic fertilizers Think of the reduction of carbon dioxide into the atmosphere from not going through that industrial process. So restoring soil biology on farms. So what I want to look at is some of the work that we've been doing at the Celebration Farm. I talked about this a little bit last time, but just to remind you all that we've got these experiments going on the farm where we're trying to figure out what is the easiest, fastest way, least expensive for the biology to get into the soil and under different conditions. So we bought a farm that the people before us were just crazy about applying herbicides, about applying different kinds of toxic chemicals, putting out, we think, massive amounts of inorganic fertilizer as well. And so when we look at the soil, to begin, the only biology left are the bacteria, and not very wonderful bacteria because this soil is so compacted. It's kind of scary. And so the first year, we just put biology on the surface of the soil. One of the two rows that you're looking at in front of you, the corn, had organisms added into the system, the other did not. We did put mulch on top of everything, so at least that's fair. That's the only difference is that we put biology in the left row, and we did not apply biology in the right row. And look at the difference in the growth of the corn. We had some, we had some variations in the kinds of biology we put in. I'm not going to talk about that right now. But you can see that difference right from the very beginning, even if you're only putting this biology in on the surface of the soil. But I would like higher yields than this. So the next year, in this part of our property, we tilled the compost in the fall. And then we tilled more compost in the spring. And then we planted the beds. And so I should say the compost was put in on the plus biology side. On the no biology side, we put mulch in. So we're trying to be fair about the nutrient inputs. We're certainly putting in as much nutrient into these soils as you would add with inorganic fertilizers in a conventional system. So the nitrogen is being added. It's just not in a soluble form, it's in the form of plant material here. So when you look at the no added biology side, you're looking at all these different plants are indicated along the right-hand side. And you can see that the corn is just tasseling out. The tomatoes, you can see right through the tomatoes to the corn behind it, so the tomatoes aren't doing really well. The okra is not really taken off at all. You can see that probably 4, maybe 5 of the cucumber plants, but we've got 20 cucumber plants in those two, in that row, because we've got a row lower and a row higher within that bed. And the peppers, yes, they are starting to produce peppers, but you can see that some of those leaves are very yellow, fertility problems. Now we compare this production to where we added the compost into the soil, where we put compost in the springtime. Same amount of work on both sides, because over there we put mulch, over here we put compost with the biology in it. And you can see how much better all of the plants are growing. You can't really even see through the cucumbers to the okra behind. You can't see the tomatoes at all, but the tomatoes are growing about as tall as the corn is growing. Every single plant of corn has tasseled. When you go to yields, on each of those corn stalks in the plus biology side, we had 3 to 5 ears of corn on each stalk. When we go back to the no biology side, we had 1 ear of corn per tassel or per stalk. Not only was it slower to develop, but it didn't produce as much. And I'll go through numbers for you in just a minute. So you can see the difference in the health of these plants because the soil was healthier. As we keep improving this biology, things are just going to get better. So can we feed the world? Absolutely we can. We don't need all those toxic chemicals going out there where you can't walk in your field, you can't walk in your garden, you can't walk on your lawn after those chemicals are applied. To just kind of do a close-up, this is the okra. We try to grow cover plants on the surfaces of all of our soil because that helps hold water. It prevents weeds. And even our cover plants are not doing well, where we've added mulch. We've put no biology on here. It's just the biology that was in this dirt before we started. The plants are eking out an existence, but this is really not what you would want your garden to look like. And yet, just on the other side of the pathway where we have added that biology through putting the compost, tilling down to 2.5 feet before we start the crops, getting those organisms into the soil and starting to see an improvement in the biology in that soil, and then applying the compost extract to the surfaces through the growing season. I believe we apply them 3 times through the growing season. On either side, if we saw pests or diseases, we would apply chemicals. On the no biology side, we would put biology. We would use a compost tea to reestablish the beneficial organisms on the surfaces of these plants so that the pests, the aphids, the white flies, the caterpillars would be dealt with by the biology in our compost that we extracted and put into a liquid form. And you can see the cover plants. So we don't have to water as much over here. When you go out midday and you stick your fingers into those cover plants, there's still moisture on the surface of the soil. You go back over here and there's no moisture. Even though we're irrigating, we've got a drip irrigation system in here, that water dries so rapidly from the surface. Yeah, it's going to stay down in the soil, but then evaporation is going to take care of that water pretty rapidly. So there are all these issues that we can deal with when we put biology into the system. Now this year we put in an irrigation. We started about 2 more acres worth of land, putting it into this no added biology, and then we started some conventional sections so we would have that comparison to what goes on with fertilizer. And we put in an irrigation system, and that irrigation system did not work. We had problems with them. So this year has been a little bit of a bust for us in terms of developing another year's worth of data. It's going to have to wait till next year because we have the irrigation system in hand now, but it's a little late to be planting things. We did an interesting experiment in the greenhouse where we started the plants that were going to go out, say like the tomatoes, some of the lettuce, some of the different kinds of plants. We started in the greenhouse. We knew that we wanted to have chemically fertilized starts because that's what would go into the conventional plots. And so our starting mix was potting soil we obtained from a commercial source, and we mixed all the right inorganic fertilizers into that potting mix along with the perlite, so totally chemical recommendations, and then started the seeds in that set of trays for the conventional plot. For the no biology, we just took potting soil and put in the rice hulls, so a slightly different form of, an organic form of fluffing agent, rice hulls instead of the vermiculite. And whatever biology was in the compost, or was whatever biology was on the rice hulls and on the potting mix that we used, that's the biology was very low, very, just bacteria. So we put our seeds in, we water, and got those seeds going on the same day as the conventional. On the same day, we then took our compost mixed in the rice hulls, because in those little trays, you got to improve airflow, so you need that fluffing agent. So we had the compost mixed with the rice hulls, and then we put seeds in. All of those started on the same day, and what we noticed in a week's time, after everything had germinated, in the chemically grown trays, those plants were only about 1/4 inch high. Where we had no added biology, those plants were about 1/2 inch to 3/4 inch tall. And in the plus biology, those starts were 1 to 2 inches tall. That kind of quickness, that ability to grow, was mimicked through the whole shortened, as it was, growing season, because we couldn't get them out until we had the irrigation system in. But that same differential in plant production has shown through the rest of this growing season. So just that interesting part of the experiment, this change in the way that plant is responding because it has biology in the root system, starts in the greenhouse. So even there, we need to be growing our starts with compost in that potting mix. Now, this is weeding required. You'll notice, there are very few weeds in that row with the okra, or you look over to the tomatoes on the right-hand side and the cucumbers on the left-hand side, I believe. There aren't any weeds. So when you look then at the no added biology, there are weeds over there, and you can see them, and we're going to have to go in and pull them. So we wanted to do an experiment, and how do you start out with both sides even? So we would go into these rows, and we would weed absolutely everything that we could find on both sides. No weeds left after we weeded really intensively. And we then came back in 2 weeks, and we then timed how long it took to weed the no biology added. So in the beet trial, with the no biology added, it took us 30 minutes. In the plus biology side, 10 minutes. With the corn, with the cucumber, you can see the differences in weeding time. And weeding is not my favorite thing to do. I would much rather be able to just quickly walk down a row and pull the 1 or 2 weeds that have snuck into kind of bare patches between the cover plants, as opposed to spending 65 minutes having to pull out everything that's grown in the last 2 weeks. And that's going to keep happening through the whole entire growing season. You're going to have to be spending that time weeding. So if you would really like to stop spending so much time weeding, let's get the biology balanced to grow the crop that you want, and select against the growth of all those early successional weeds. I can show you many pictures like this where people will put in a crop, and yes, this is organic production area, so all of this was organic. The back area, they just continued with the normal organic practices, the sprays and the organic fertilizers going out. And this front was sprayed once with 1 tea application. And look at how that chard has exploded in growth as that good biology got into soil that was already not bad, because it was managed organically. Most of us wouldn't have any problem with the chard in the background of this picture, but it would be wonderful to have the growth that we're seeing in the front end. So Daniel and Caitlin MacLeod did this work, and they were really happy, so they sent the picture, said that we could use it, and this is the kind of result that we see time and time again. if we go into the soil and we measure the biology in the front part versus the back part, what makes the difference is the fungi have come along. We've applied a compost tea that had real good levels of fungi in it, also some protozoa, also some good guy nematodes, and look at the difference that it makes, even in something that is a well cared for organic garden. Another example. Yep.
Do a quick time check for you on slide 21 out of 43, and we're about 55 minutes in.
Okay, I will speed myself up. Okay, thank you. Okay, so going on with an onion grower, this is out of New Zealand. So Tasmania in New Zealand. Sorry, Australia. Duh. This is Australia. And the grower said, conventional for many years, and he wanted to see if all the fuss about biology was real. So came to us and he said, would like to trial biology in some of his fields and no biology in others. And so working with one of the local Soil Food Web advisors was making very good compost. So see, this is where we were lucky. There was a person close by who could already had the biology that could just be used for these trials. So this is the conventional system. Look at all the weeds in there. This is even after 2 applications of herbicide. I don't remember which herbicides he used, but they were standard, typical ones to deal with these particular thistle problems. Even when I look at this paddock, this field, I often go, where are the onions? It's been 2 weeks since these onions were planted as seed, so yeah, they should be just coming up. And it's really hard to see the onions when you look across this field, except for where you look right down the row. So you can see those onions here in the foreground, but it's hard to see the onions when you look. You can see the weeds, can't you? Yeah. Now we're going to be looking a little bit closer at where we dug up some of the plants in just a minute, but let's look at the plus biology side. So an application of really good compost extract going out onto this soil, so the biology was added. Yes, this is Tasmania, so we've got good moisture. We don't really have a dry time of the year. So springtime, moisture, water, rain, moving a lot of those organisms deeper into the soil, and there are no weeds. This was a field that had as much weed seed bank in it as the previous field. The grower said, oh no, he's always had problems in this field with the weeds, always. And so this soon, after moving along with the biology, he was convinced that he wants to go biological now. Well, he let us finish the trial, and he did get higher yields with the onions from the biological fields. So another reason is look at the structure of the soil. This is, again, the conventional system. Look at all the weeds. The onion seeds have just germinated and started to grow. But look at the structure in the soil. There's some earthworms that have been born through there, but that's it. Would you want to put your root systems in here? And when you look at this little seedling right here, notice where there's already disease attacking that root system. We already have a problem starting to develop on the roots of these plants in the conventional side. He is going to have to use some pesticides to try to deal with these problems so he can get a yield of his onions. Look at the biological field. Look at the structure in here. Already you can see that there are lots of air passageways and hallways. There are so many aggregates that have already been built. The earthworms are going crazy. And look at the root systems. We had to go further back away so we could get all of that above ground part of the plant. It's a good inch and a half taller than the conventionally grown plants. And those root systems are going down a full foot and a half, even this early in the growing season. Whereas you go back to this one, that's if you're lucky. that's down to 6 inches. So it's going to make a big difference in yield. We've looked at grapevines, also in Australia. So this is up in the middle of the country, in some of the more productive wine grape growing. These are table grapes, not wine grapes. Working with the Grape Exchange in Australia, they gave us one non-productive farm. So in 1 year, we started normal yields in the middle of a drought by putting compost along the beds where the grapevines were coming out, 2 applications of compost tea going on after that. But you have to make certain the biology is in the compost. It's in the compost extracts or compost teas that you're putting out. And so in that first year, 1 growing season, we were the only farm in Australia that was growing table grapes because it was in the middle of a drought. The big retaining areas, the water retention areas on this farm were dry when we started in the growing season. In the springtime, we got barely any rain at all. That water retention area never had water in it through that whole entire growing season. And yet, because we got the biology established in the soil, the roots of the grapevine started growing deeper into the soil. They got to some of the water that was actually present deeper in the soil. And one of the things we noticed was that dew was very important. Those days where there was actual dew fall, all the dew infiltrated into the ground, and that was now water available for the plants to grow. And it was enough to allow yield, normal yields in that first year, even though so much drought was causing everybody else's crops to fail. In 2007, all of the farms belonging to the Grape Exchange Australia converted. They weathered, again, severe drought. In Australia, I think they've been in severe drought for the last 35 years. I think it's climate change. I don't think that's drought anymore. But all of the farms were increasing yields as compared to the farms that were left conventional. They reduced operating expenditure because you don't have to have people out there putting on inorganic fertilizers and pesticides and picking bugs off your grapes and all of those kinds of things. So, you know, large scale operations, yes, we do that. Barley, here's another example for those of you. Hopefully you're not getting tired of this yet. I was brought on board because they were having poor harvests. They were trying to go organic. And we said, well, yeah, but even in organic, you've got to make certain that you get the biology in there. It's going to take you 5 years to go through the transition. And really, you want to go through that transition in the first growing season. So they did an experiment, so a control area with no treatments. So just the standard chemical applications. Then an area where the seeds, were coated with compost extract. The third treatment was where the seeds were coated. Well, there's a few typos in here. Were coated with compost extract, and then the plants were we applied compost tea when the seedlings were very small. So when they had first popped up, first true leaf stage, go out and apply a compost tea to the foliage. And of course, you're going to get the ground at the same time. So with enough moisture, the biology would move into the ground. And then the fourth treatment was we coated with compost extract on the seeds, and then 2 applications of the compost tea going on the plants. Everything had been treated with organic fertilizers in the last couple years. So, you know, there was organic fertilizers put on. We made certain that we had really good biology in the compost, really high diversity. We had a fungal biomass that was upwards of 500 to 600 micrograms of fungal biomass. and only about 300 micrograms of bacterial biomass. So this was fantastic compost. This was exactly what we wanted. So when you look at the numbers of tillers, you can see that the average number of tillers increases with more biology, moving the food web in the right direction. You can see with number of seeds per seed head increased. So each of these tillers produced their 1.6 tillers per plant. So the head had 30 seeds. But now you'll have almost 3 tillers per plant. Each of those tillers produced a seed head, each of which had 32 seeds. So we even more than tripled yield here with just an application of the proper biology on the seed. But you got to have really good compost. That's the part that you have to have. Here we are where with applying a application at the first true leaf stage of the biology, that we have 6 tillers, and each of those tillers on average produced a seed head with 34 seeds. Here we had 8.5 tillers. So each of those heads on each of those tillers had 36 seeds. So we've more than 100% increase in yield with the full complement of applications. You can see that the weeds decreased significantly, and the weeds were short. They were unhappy, they were unhealthy in these treatments. Well, they were fully healthy. And so of course you're not going to get as much yield. Compaction went down. The compaction layer was right up at 4 to 5 centimeters. So that's just a couple inches below the surface to we're down a good foot or so into the soil where the root systems first encountered a compaction layer. And so better root systems don't have to fight to push their way through the soil. So these results are very common as long as you're getting the biology in the compost and then transferring that compost biology into the soil. It's where people don't get the biology established in the compost. They're applying, tons of compost goes out there and they only see a little bit of increase. But they didn't put the biology out. So, come on. You've got to follow all the instructions. So in this case, in that last treatment, 500% increase in yield. So photographs of the actual crop. So this is with no added biology at all, just applying to the seed, and you can see the increase in tillers, applying it to the seed and to the first true leaf stage of the plant, and then applying to the seed, and then 2 applications, one first true leaf stage and another at about a month later. So what kind of crop do you want to be growing? I know what I want. This is from a peanut farm in Georgia. So Texas Organic Peanut Farm, sorry, from Texas. We have other people in Georgia that we work with, but here's another. So the increase in yield between 2015 and 2016 was 20% increase with just 2 compost tea applications. So making his compost, learning how to extract the organisms from that compost and then grow them, making the tea application. The problems that they had, of course, were recurring drought, conventional farmers losing money. 2016 was the 3rd warmest year on record in Texas, and instead of increasing his fertilizer use, he opted to restore his biology. So 5 gallons of compost tea were applied, and please recognize that because these are peanuts and they should be mycorrhizal and they are leguminous, both the mycorrhizal fungi and the rhizobium went into the tea, so that we grew the rhizobium in the tea during the brew cycle. The mycorrhizae were applied just before we were put into the tea, just before we applied the tea to the planting. So they checked the biology, and the organisms had increased dramatically. They did a pretty good job of wiping out the root-feeding nematodes. So when we're looking at some nematodes being present, mainly bacterial feeders, a few predatory nematodes, but they reduced the root-feeding nematode population almost completely. So the difference, this is what the crop looked on July 23rd in 2015 as compared to July 21st in 2016 with very similar weather conditions, just dry, 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 dry. So again, which crop do you want to be growing? You get closure of the rows here and you don't have weed problems. You help maintain moisture underneath those plants as they get bigger and prevent evaporation from the surface of the soil. So all kinds of benefits. All the biology, all over all of the surfaces, reduced diseases, reduced pests, so 20% increase in yields. So compare tonnage, 225 tons versus 270, $54,000 increase in profit. And that's the increase in profit. So even on large acreages, it's possible to get the biology out. We have to make certain that the biology is in the compost. You can make small piles or you can make ginormous piles. You've just got to manage it properly. And that's where the advisors come in, very important to get that help. Somebody coming out and actually testing your compost to make sure that it's ending up with the improvement in biology. We're seeing that in some places in the United States there has been so much application of pesticides, inorganic fertilizers, toxic materials, that there is no life left. We're dealing with practically sterile conditions. The produce being made, even on the surfaces of the residues, there is no biology on the surfaces. So if you take those residues to make your compost, you're not getting the inoculum. So it's really critical to have somebody helping you with the biology, making sure that the organisms are growing in your compost, you're producing what you want. So weeds, soil erosion, nutrient deficiencies, pests, very doable. We're going to go into a section about compost, and I've got 10 minutes to do this. So I want to just first of all go through a very typical scenario with numbers of these organisms, so you kind of get an idea of what we want in that compost. First of all, we're going to look at an agricultural field. We always go out and take samples from the soils before we start. Each growing season, each fall, we want to be knowing what has actually happened. What are we starting with? So this field is, what are we starting with? And this is 2 weeks after we added this compost. And we're going to look at total bacteria. We were doing metagenomics on these populations in the soil and in the compost. And so we're looking at the number of bacterial species per gram of soil using a DNA analysis. We're looking at the microscope assessment of total fungi, and then the number of fungal species using a DNA approach. The protozoa, we're looking at flagellates, amoebae, and ciliates. The flagellates and the amoebae are the good guy protozoa that eat your bacteria, releasing nutrients right in the root zone available to your plant. Ciliates, the C, the last number in this grouping of 3, those ciliates are indicative of compacted anaerobic conditions that allow the disease-causing pest species to grow, attack your root system, and reduce your yields. So we don't like seeing ciliates. That means something very, very bad. So let's go through the agricultural field before we started. 300 micrograms of bacteria, that's quite good. 5,000 species. That means we have a lot of individuals of a very few set of species in that soil. This is not nearly the diversity that you have to have in order to suppress disease in that root zone. It's not nearly the number of species of bacteria that we have to have in order for your plant, when it puts out specific exudates, it's expecting to find these very specific bacterial species to grow and do this job. With only 5,000 species of bacteria in your soil, there is not a prayer that when that plant puts out the exudates, that the plant is going to be able to get back the energy expenditure that it put out by putting out that exudate. Nobody's home to help the plant. Going to the fungi, this is just so low. We need minimum 300 micrograms in hopes of having a decent crop in most conditions. And so 5 is just way, way, way too low. Going down to the number of species, way too low, even worse than the bacterial condition. So when your plant's putting out those exudates, so much of the time it's not getting any benefit. Plants, I'm knocking on the door and nothing's happening. Flagellates, absolutely none. Amoebae, there is no nutrient cycling in this soil. Well, it's not soil, is it? It's dirt. Because look at the anaerobic indicator. The diseases and pests in here are going to be painful. And just try to kill them all with toxic chemicals. It's not going to work. The pests have already become resistant to everything we're trying to throw at them. So how do you fix this problem? You build the organisms. So let's look down the compost layer. We applied compost at 1 ton per acre, but we had really good biology. Yeah, the bacteria were a little high, 2,000, but look at the protozoa. That's going to start to knock those high bacterial numbers down. So, just hold on. The protozoa will do their job. But we have 75,000 species of bacteria in that compost. If we use that to inoculate your soil, we're going to have all of the organisms, all the species present, that when the plant puts out those exudates and is looking for somebody to do the work for that plant, somebody's going to be there to do it. No matter what the weather, no matter the temperature, the moisture, the calcium concentration, whatever, salts, blah, 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 blah. The organisms are there to help that plant. 950 micrograms of fungi, well above the 300 microgram minimum. So yes, we are getting 25,000 species of fungi. Yahoo! Great compost. That's what we want to be seeing. If you're looking at a compost that has 15,000 bacteria and 50 fungi, is it worth it putting anything out using that? Well, okay, putting out some bacterial diversity would still give you some benefit, but it just might not do everything that we've been talking about today. We got the protozoa, we've got the flagellates, we got the amoebae, the ciliates are down way below a level of concern, so this is good aerobic compost. Applied at 1 ton per acre. 2 weeks later, how much has this application of compost improved the biology in your soil? Way above the 300. So yeah, it's a tiny bit on the bacterial dominated side, but certainly for most of our vegetable crops is going to result in all the benefits happening. We got good protozoa, we've got good diversity. Notice that if we put the inoculum in the compost, all of those organisms are spreading throughout all of that soil and getting the diversity up to where we need it to be. So that's what we're trying to do with compost. So when you're making compost, you want to make certain that the starting materials you're putting in there are going to be such that it's going to grow those organisms, get them growing so fast that they will raise the temperature above 131 for at least 3 times. You've got to move what was on the surface of this, everything on the surface of that compost, top, sides, and bottom, aren't getting above 131 because they're in contact with ambient outside temperature. That means we're going to have to turn this pile so that all this outside material is going to get into the inside and it's got to get hot enough long enough. So we're using thermometers, taking the temperature of the pile, making sure that it's above 131 for 3 days. And that will kill all the pests, the pathogens, the parasites, all the bad stuff. So no, there will be no disease coming through, no detectable E. coli, no detectable fecal coliforms of the bad variety. So got to do that, and so turning. So here's our turner. It's a self-propelled, kind of small size, because we only have a 60-acre farm, so we don't really need more compost than making 1 to 2 windrows every month through the whole entire year. Well, we usually take January, February, March off. It's too wet. But after we get our permanent composting site, we will be making compost year-round. So as you turn this, what's in the middle gets thrown up. You can see it being thrown up to the top and spreading to the sides. What was in the this top drops down to the sides. What was in the middle gets pulled into the center. And so 3 turns. If you do this properly, 3 turns is all you need to make certain that all parts of this compost pile get into the middle. Well, if you can do 4 turns, if you can do 5 turns where you've been above 131 degrees Fahrenheit for a long enough period of time to kill all the pathogens and the pests, then it's just icing on the cake. It just makes more and more certain that you don't have any pests or pathogens or diseases. Keep it good and aerobic, because remember, the pathogens grow in reduced oxygen conditions. They cannot outcompete the aerobic organisms. And I explain why in the full course, the Life in the Soil classes. So another composting operation, people tell me that you just can't make that much compost. Want to bet? We work with growers in South Africa that make enough compost to be putting out 10 tons per acre on all of their 300,000 hectares of land. It's totally possible. You got to have 300,000 hectares of land to have enough waste material to make this much compost, but it's entirely possible. So you've got to have a turner, and so again, we see a turner, large scale like this. You've got to have a water wagon, so you're keeping the compost at 50% moisture. You want it right about that level. If it starts dropping down to 45% moisture, you want to get your composter turner out the next time you're going to turn your pile, and you're going to add water. And notice that the water is being sprayed out of nozzles that come into this pipe at the top of the turner and spray out, so that as the compost is turning, each and every tiny little particle is getting coated with water. And that's the best way to wet up compost. So large scale, this particular composting operation that I was being brought into by a helicopter because it was so far away from everything. As we're flying towards this composting operation, my heart's just dropping in terror, because look at the black plastic covering these piles, and look that somebody had actually put dirt over the sides of that black plastic, so it was completely sealed. This was anaerobic. The disease-causing organisms growing in the when we're with the biology and who you want and when you want and how to grow them and all of these details. And then we have a compost class that we go into the gory details of making compost. And then another class that is on making compost tea and compost extracts. And if you don't know what the difference are between those two things, you have to take the course. And then finally, a course on how to use the microscope to do the assessments of all of these organisms. You can't do anything if you can't measure what you're doing. And so you need to have somebody in your organization that learns to use the microscope, or you want a friendly microscope person close to you. So you can take all the samples to them, have them run them, and within minutes know the answers of what you need to do to continue fixing the problem. So hopefully that was interesting to everybody. I can go on for about another 30 days talking nonstop about this stuff, but it's a good place to stop and let Vivian have a chance to take over.
Sweet.